coming to seminar, especially on Friday before Labor Day, heading into the long weekend. So it's great to see a nice full house here. If I haven't met everyone here yet, my name is Steve Devlin. I'm the director of the Masters in Data Science program here at USF. Lots of our students are here. This is the second seminar of the year for us, so we're excited to keep seminar rolling along here. So I think we'll more or less jump straight in. A couple of quick announcements. The certificate courses run through the Data Institute are starting soon in the beginning of September, which is days away. So you can check online for the Data Institute website to find all that, and that's also a great place to check in just to keep up to speed on all the events that are happening through the seminar and the Applied Data Ethics Center as well. Okay, so we're gonna jump in for today's talk. So we're excited to have Neal Grantham here. Neal is coming from Phylagen, so for the students this is a quick practicum preview, so pay close attention. And Neal's got a Ph.D. in statistics from NC State, and like any good statistician, has a math background before that. So we're happy to have him here. He's going to talk to us about data science at Phylagen. Thanks, Neal.
Cool. Thank you. Haven't even done anything yet. All right. What's this? Okay. All right. Thank you. And thanks for having me. I'm excited to be here. It's been a little while since I've given a kind of seminar talk, so I think I'll get back into the rhythm, but there'll be potentially a rocky start here. So I'm here today to talk about data science at Phylagen. First of all, this thing's not working. There we go. Who am I? My name is Neal Grantham. I have a Ph.D. in statistics from NC State. It was 2017, still fairly recent. Grad school memories are still alive. Before that, I was at Cal Poly in San Luis Obispo on the Central Coast. That's where I did math and stats. So obviously, eventually I migrated over into data science. When I was there, I researched Bayesian spatial models with Brian Rich. One of my first projects was on air pollution modeling. And I was a young grad student, and I was too afraid to tell my advisor that I absolutely despised that project. It was this, like, trying to squeeze blood out of stones, like trying to get like epsilon lower mean squared error on predicting pollution across the southeast US. But also as a great advisor, he kind of picked up on my disinterest, and there was a meeting one time where he was saying, hey, I got this email from some people in the biology department, and they have this really interesting data set that they're wondering if somebody with a stats background can work on, right? And tell me more. And it was this data set of, the project is called Wildlife of Our Homes, and it's this data set of citizen scientists, members of the public who participated in this. These members at NC State, Rob Dunn, Holly Menninger, and Noah Fier at CU Boulder, they sent swabs, just kind of sterile swabs to people's homes, and asked them to collect dust, essentially, around their home in certain areas, right? One of those areas was the outer door frame, like facing the outer elements, right? And in particularly in areas that are not regularly cleaned, right? And so they got all these samples back, and what they did is they put them through this high-throughput DNA sequencing, and they identified tons of different bacteria and fungi and pollen that are all present in these particular samples, and they used that to kind of characterize what kind of microbes people are exposed to in their day-to-day life on a daily basis. But they had a hunch that there was something more you could do with it. And so what I worked on post-pollution as a much happier but still challenged grad student. Is there a way to get this? Is it my computers? Okay, yeah, I can do that. Which one is that one? No, I think it's this one. I told you it would be a rocky start, okay? How's that? Oh my God. I thought I was projecting well enough, but this is nice. I still have some more slides to go. Anyway, so I got started working on this project, and what I tried to do was use the microbiome data to essentially, is this working now? Great, okay. Now we're in business. Okay, I used the microbiome data to essentially characterize particular regions and predicted, if you had a sample that you weren't sure where it was from, like let's say it was collected from a package that arrived and you have no idea where it was sent from, is the dust that collects on that package indicative of where it might have come from, right? That was kind of the loose question I was trying to answer. So I worked on statistical methods for high-dimensional, spatially distributed microbiome data from next-generation sequencing. And then, of course, on the last 6 months of PhD program, I'm wondering what's next. I knew, kind of had a sense that academia wasn't for me, so I was looking at industry, and the novelty of the microbiome data was fun to work on, but I was like, I don't know too many companies that are doing that. Lo and behold, I kind of searched around and found this company in San Francisco called Phylagen that was enormously vague on what they were doing online, but it looked like it was environmental microbiome stuff. So I reached out, we had some conversations. It turns out that we were both good fits for each other. And so I joined Phylagen in October 2017. So I've been here almost 2 years, been able to work on some cool stuff. So a little bit continuing kind of the research I was doing, but certainly on different applications. This is the team. When I joined, I was like 9 or 10. Now we're 20. The company is started by Jessica Green and Harrison Dillon. Jessica was a professor at University of Oregon, where she worked on indoor environmental microbiomes. And Harrison Dillon is a biotech entrepreneur who had a biofuel company that eventually reached IPO, and he was kind of looking for his next thing, right? And they met at a TED Talk. He really liked Jessica's talk, and they decided to start a company out of it. So now we're grown. We've got the business development and commercial ops team. We've got a pretty large data science division. A lot of you might know Henry Humadi. He's my new boss. He started about a month ago. He's why I'm here. And then we also have a laboratory team down at the bottom that preps all these different samples. So we're a little, we've got kind of a diverse mix of people here. So I said environmental microbiome. What does that mean? With apologies to the germaphobes in the audience, everything, everywhere is covered in microbes, especially the human body. There were some estimates that, there are more bacteria cells on your body than there are human cells. Original estimates were like 10 times. It actually turns out it's more like 2 to 1 or 1 to 1. But we are covered in microbes. We have tons of microbes in our gut. And there's also microbes everywhere. Anywhere that there's dust, even when you can't see dust, there are microbes. So these things are everywhere. In particular, bacteria, fungi, pollen from plants. They collect on these surfaces, and they leave kind of traces of their genetic material. Their DNA is there, right? So even if there's no actual living bacteria on a surface, it's very likely that there's like a kind of like a bacteria DNA graveyard there. And it used to be the case way back when, where if you wanted to identify these bacteria, you'd collect the sample, you'd put it on a petri dish, you'd wait for a while, it would grow, and then you'd take the petri dish and you put it on a microscope and you'd see what was there, right? Fortunately, that takes time. It also gives you a biased picture of what's actually there, because it's only the stuff that would grow under those conditions is what you're going to observe. And also, you're looking by eye, right? There might be some more automated methods, but it's not a very data-driven approach. So it used to be how it is, but now, no more. We can do a much more data-driven approach thanks to high-throughput sequencing. That was in my dissertation title, was next-generation sequencing. Now that's current-generation sequencing, so everyone's been calling it high-throughput sequencing. It's enormously more affordable now. So this is a graph of the cost associated with sequencing a million nucleotide bases. Back in 2001, it was close to $10,000. There's this Moore's law projection, which it followed quite a bit until around 2007, when these high-throughput sequencers became much more widely available, and the cost absolutely plummeted. I'm not sure what the reason is for the second smaller plummet in like early 2015, but the point is today this is much more affordable to get these very, very, very high dimensional deep views of the bacteria and fungi and plants and stuff that are in a particular dust sample. So here's kind of how the process goes. As you might expect, you just collect a dust sample. Sometimes it doesn't look that dirty, actually most of the time. You'll collect something and it looks like you didn't do a good job, but it's there. There are microbes inside of that dust, and then inside those microbes is their DNA. This goes through a very, to me, very complicated lab process. The joke at Phylagen is if the data doesn't look good, everyone says, who let Neal in the lab? Because I have no wet lab experience at all. So I arrived at the scene at the very end of this pipeline. And so you go through, you prep the data, it gets put on the sequencer, it does this really cool chemistry stuff that I don't know quite how it works, and then it identifies all the, the different sequences of DNA that were present in these particular samples. Now, you don't put on one sample at a time. You typically put on 200, 500 samples at a particular time. And what that results in is you get about 150 megabit text file of DNA sequences per swab. Right? So you can compress that, which is nice, but it gets pretty big pretty quickly. Just to give you some numbers on how diverse these particular samples are, these numbers vary depending on what kind of sample data you're looking at, but typically we get somewhere on the order of 50,000 different types of bacteria, 20,000 different types of fungi, 5,000 plants. And this data is not ready to go. It's not ready for analysis at this stage. There's a whole lot more extensive data processing that has to happen. My coworker Simon has a lot of experience kind of getting this raw genetic data and getting it in a form that's much more ready for machine learning analysis. So that's a whole separate part of the pipeline that I'm not going to talk about. But essentially what you get at the end is for each of these different samples, again, this is simplified, but at the end you get this like microbial composition, right? Which is, in this particular sample, these are the different types of bacterial species or fungal species that were observed in the particular sample and their relative proportions with respect to everything else in that sample, right? And so that leads to these very colorful, nice-looking plots. So each of these different rows is a different sample, and so for example, that first row, this is the different kind of associated groups of bacteria, of fungi, of plants. And these bins are not species. I can't remember what taxonomic rank they are, but the idea is that they are kind of, within each of those colored blocks is a whole collection of different species, but they're grouped based off of how phylogenetically related they are, right? So each color here represents a bunch of different species. This thing's freaking me out over here. Okay, here we go. And so what that means is in these samples, we detect bacteria and fungi and plants. And the great thing is these are not just random assortments of things, right? And it's not like they're ubiquitous. So if I took a sample here and I took a sample over in Oakland, it's very possible they look quite different. And that's actually repeatable. If I collect samples, a lot of samples in San Francisco and a lot of samples in Oakland, I can get a pretty good view of what Oakland microbiome looks like or what a San Francisco microbiome looks like, right? Here's a particular example. This is just one species, but this is the stinging nettle plant. On the left is a map of where it is known to occur, right? And this is from some plant database. So we see that it's predominantly in the north, and it is not really present in the south. On the right here are some samples that we took in a particular project across the north and across the south, and kind of how abundant the stinging nettle DNA was in those particular samples. So as we see here on the left, it's not present in every sample, and in some samples it's not super present, but it does have a much higher probability of occurrence in the northern samples than the southern samples, right? So essentially what we're saying here is that the microbiome data and geography are related in some way, right? And this has been known for a little while. This is a quote. I do like the quote, particularly the end, so I'm just going to read it. This is a French researcher named Edmond Locard. He said, "Among recent researches, the analysis of dust has appeared as one of the newest and most surprising. Yet, upon reflection, one is astonished that it has been necessary to wait until this late day for so simple an idea to be applied as the collecting, in the dust of garments, of the evidence of the objects rubbed against and the contacts which a suspected person may have undergone. For the microscopic debris that cover our clothes and bodies are the mute witnesses, sure and faithful, of all of our movements and of all of our encounters." So he's talking more in the kind of forensic sense. It's kind of very like Sherlock Holmesian kind of view of using dust, right? And he says, it's such a shame that we waited until this late day to explore this stuff. He wrote this in 1929. So it's taken quite a little bit of time to get to this stage where we actually have the technology and resources to realize Locard's vision. I'm not sure they were ever able to use dust in any particular sense in his capacity as I think he might have been a French police chief, I'm pretty sure. But I think it's funny, this kind of the idea of dust having extra information in it has been around for a little while. So this is an old idea, but we have, thankfully, some new tools available to us that can make it a much more data-driven approach, right? And so the idea is to use this microscopic debris as a proxy for geographic information. Kind of the major steps are we want to identify the DNA that's present. That was that whole kind of pipeline I walked through. And then this is where the kind of data science stats comes into it. We want to train some models to essentially, based off of different geographies, detect which microbes and stuff are indicative of particular regions and not of others, right? And the idea is if we can build these models that are really good at looking at microbes and predicting geography, we can use that on samples and other situations where we don't have the geographic information available and we want to use this dust that collects on everything as kind of a vehicle to make some sort of prediction, right? So switching gears just a little bit, I told you guys started in July, so I didn't want to like jump right into machine learning, but to give you kind of a useful example to think about in terms of machine learning, how do data scientists train computers to recognize handwritten numbers? Well, it starts off, you have a big giant data set of handwritten numbers there, and you know which ones are ones, which ones are twos, which ones are threes, et cetera, et cetera. And then you choose a particular machine learning model. That might be a classical model like random forests, or more recently, neural networks. And you essentially show the particular model, the data, and what the labels are, and it essentially turns its knobs and dials and tries to figure out essentially the best way to predict, based off of the pixels in the image, what the image actually represents, right? And so this is an example of you're showing it an 8, and hopefully it kind of passes through the available nodes and layers and kind of results in a strong prediction of 8, right? And the way we gauge the model and test its performance is we have a holdout data set of numbers that we know the labels on, but it doesn't know, and we let it predict and kind of see how well it does, right? And so in this case over here, there's 50 different numbers. It gets 3 of them wrong, 2 of which I would probably get wrong. That 2 definitely looks like an 8, or it looks like a 2. I don't know. So we can gauge the model's performance and then kind of tune appropriately, right? So this is a general idea. You have images, they represent numbers, and they're composed of pixels, which is kind of what the model is using, right? Now, ideally, the model would use the structure of the pixels, but I'm not going to super get into that. Relating this to the microbiome, just so we're all on the same page, you can think of a single image as a single dust swab, right? You could think of the pixels in that particular image as the different genetic features that we saw. Maybe the white pixels are ones where we observed really high abundance of them, and the dark pixels are ones where we didn't. Now here, they have some sort of spatial structure. 2 pixels are close by one another. We also have structure, which is the phylogenetic structure, right? We know that certain bacteria are more related to others. And so we can kind of incorporate that information as well. So we have structures, it's not quite this spatial structure, right? And then finally, the number, the thing that the image represents, to us could be the geographic origin, right? Now that could be a particular building, it could be a particular city, it could be particular longitude and latitude coordinates. It depends on the application. But the idea here is we have swabs, they're like images. The pixels are the genetic features, the microbes, and the label is where it's from, right? So I'm going to walk through. I would love to keep going further and further into how the model works, but instead I'm going to talk about kind of like what it is that we're working on, right? And how are we using this microbiome data in different applications, right? So one particular place that we're applying this technology is in the supply chain. Typically what happens is if you're a brand in the US, you get your stuff manufactured overseas. You enter into these contracts, and then at some point in the future, a whole bunch of products arrive in the US. that you asked a particular facility to manufacture, but you don't have any sort of visibility into what happened in between you entering that contract with this company and the goods arriving in the US. Now, they might meet certain quality standards, but there's a whole host of things that could happen in between there, right? In 2013, there's this Rana Plaza collapse in Bangladesh. It killed over 1,000 people, and it was this facility that was doing manufacturing, but they were not authorized to be doing the manufacturing. So what had happened is major US brands had entered into contracts with companies abroad, and those companies had essentially outsourced the manufacturing to places that don't meet certain safety standards or ethical standards, and to make a little bit more profit. And so these particular regions are not examined so closely, and in this case, disaster struck. So what we would prefer to do is we would prefer to verify that when you enter in a contract with a company abroad, that they are kind of appropriately following that contract. If they have a certain set of facilities, we would prefer to verify that the goods that we enter into the contract with are manufactured in the approved facilities, right? So here's this gets a little CSI in parts, so forgive me for that. I'm not one to embellish, but I didn't create this video. Hold on, let me see. Let's see, here we go. Sorry, it's loud. I don't know how to do that. Let's see. Such is life. I don't get that kind of display, unfortunately. Oh, that's that. All right. So this is essentially the kind of supply chain offering that we are working on right now. Let's see. Here we go. Sorry, one second. This video. Okay, another possible application is transportation of goods, whether that's over land or overseas, right? Again, if we can swab the dust and it tells us something about where something is from, we might be able to kind of track different routes that goods took. So in this case, this was before my time at Phylagen, but somebody, very lucky individual, went to Oklahoma and had a certain amount of packages. They were not trying to swab the dust inside the particular packages. They were essentially trying to see if these packages, when they start, all have kind of the same microbiome on top of them and on the package, and as they move through these different kind of transport routes, whether or not their microbiome signature changes, essentially. So they set sets of different packages and sent them on different carriers. In this case, it was UPS, the US Postal Service, and FedEx. And before, they did not have, or they were all in common. They all kind of looked similar. And then afterward, there was these kind of clustering regions of microbiome, because the packages were exposed to different environments. They're on, on these different shipping routes, right? We can also, this is over land. We had another project where we did it overseas with shipping. So what happens is you might be shipping in goods from elsewhere, and the boat that's carrying the goods stops in certain ports along the way. We're told it's a problem that sometimes these boats make kind of unauthorized stops in regions and pick up things that they're not exactly supposed to. And so what we did in this project is try to see what's the viability of looking at the microbiome of the ports and matching them with the microbiome of the boat based off of where it goes, right? And so what we did is we had samples from port in Honolulu, Los Angeles, Oakland, and Seattle. And then we also had these boats that were traveling different routes. And we went to the ports. These ports are large, so it was we kind of sampled quite a bit everywhere. And also the boats are quite large, not as large as the port. But we got on the boats and kind of collect dust samples went at their final destination. And then we were able to essentially match which ports the boat went to based off of the microbiome that had collected on the boat because it was associated with the port microbiome signature. So this shows, this is a simplification, but this shows these kind of different kind of genetic signature patches across these different ports, right? So this is, again, another example of using the microbiome technology to kind of like track things as they move, essentially. Kind of one that was a little different and nobody was really sure it was going to actually work was we did a pilot with a major pharmaceutical brand on counterfeit pharmaceuticals. So what happens, a major problem, is that there's a lot of pharmaceuticals out there and people make kind of counterfeit versions of them that don't do anything close to what they're supposed to and are probably quite dangerous to consume, right? And so oftentimes what happens is these major pharmaceutical brands have partnerships with local authorities, and they kind of seize these counterfeit goods when they encounter them, right? But what they're not sure about, they know that they're counterfeits. They can look at the packaging. You can do a chemical analysis. Usually they're just not super well made, right? But what they don't have information on necessarily is, essentially where are these things coming from, and are maybe two different seizures that you got in two different areas, are they from the same manufacturing origin, or were they made completely in different places, right? So this was a huge lift for our lab team. It's not, you can't just do the dust swabs. You had to figure out some way to essentially kind of like dissolve the pill and recover the microbiome from it. So that took quite a bit of R&D, but they figured it out. So this is a plot. Sorry, there's kind of a lot of information here. But the y-axis here is the number of microbiome or microbial species that were observed in a particular pill. Each point here is kind of like a single pill pack. And what we have is the different kind of facets, the different columns here are different known manufacturing origins, right? So this is kind of a little bit of a pilot engagement to see if we could kind of detect these differences based off of manufacturing origin. So what we have here, thankfully, the authentic pills have the least amount of microbial DNA present, which is great. Good win for the pharmaceutical companies. And then some of these are just absolutely riddled with different types of bacteria, right? And this doesn't necessarily mean that these are like enormously super harmful bacteria. It's just they're not supposed to be there, basically. And it's probably because these different things were, certain materials in the pills were cut with something else, or just the manufacturing process itself was not very sterile or clean. In particular, whoever was unfortunate to buy some of these ones up here, I mean, I at first thought that was a mistake based off of how much microbiome DNA we found in there, but that was a particularly dirty set of pills. But the idea here is that, first of all, we can kind of detect counterfeits by how polluted they are with bacteria. That's one. But then the other thing we want to see is, do these pill sets kind of cluster by their manufacturing origin? And so this is a plot. 2 points that are close together are more similar, and 2 points that are further away are more dissimilar, right? That's all you need to know about this clustering exercise. Again, those are the authentic pills. They have some friends there in the pink, right next to them, sorry, a little difficult to distinguish, there are kind of a lot of colors here, but there's another group here that is also pretty low in the microbial count, and the microbes that are there do look similar to the authentic ones. So what that suggests to us is maybe this is like a, like a Walter White Breaking Bad type like manufacturer of these counterfeit pills, right? It's got all the good stuff. And then you have kind of these other clustering regions, right? You've got those points at the top, you've got the points down in the bottom left, down in the bottom right, and these correspond to the manufacturing origins of these different places. Now, this doesn't necessarily tell us that, like, oh, these ones down here are from region X, unless we have good representative samples from region X, which is pretty unlikely. However, you can imagine a situation where they actually do get a seizure, they find that that is associated with these certain packages, and then they have extra information as to where that might be from. Or maybe we are able to look at the microbial features in there and associate them with kind of different regions of the world, right? This is something that we're actively working on. And then finally, last application I will talk about, I think this one is the coolest. It's also at the moment the least commercially viable. That's probably why it's so cool. But you might recognize this place. It's a little city that we all call a home. And what we did is we wanted to make a microbial map of San Francisco, right? And so what we did was every month we went to these different sites. There's these kind of two regions. We went to Pack Heights and the Chinatown area, and we also have this 1 kilometer by 1 kilometer grid across San Francisco. And our fields team, composed of Tarn there in the vest and Nasser down there in the bottom left, we drive around and basically collect dust samples from areas that are not regularly cleaned. So you'll see here, there's like that newsstand kind of up in these nooks and crannies, kind of on this light fixture here, right? Just kind of collecting areas that have just passive dust collection, right? I took this one. We have a beautiful Golden Gate Bridge in the background. Nasser's looking cool in his sunglasses. This is kind of our poster child for collecting samples. But we have these samples, and we have them every month for a year and a half, right, across this particular region, because we just wanted to kind of get an idea what's the spatial variability, what's the temporal variability, that kind of stuff. And then, so what we did is to kind of test ourselves, the kind of numeric model testing where you withhold certain points and you try to see how the model does. We trained a machine learning model to essentially kind of put a pin in a map and try to figure out, based off of the microbiome of samples that it hasn't seen before, where in San Francisco does it think that sample might have come from. Here's a collection of different ones. This is something we've shown to investors, which is why we have the nice little major sites of San Francisco there, just in case they're not familiar with the city. But you should be focusing on the kind of points and the markers there. So for each marker is a true location of where a swab was collected. And then the points of the associated color are essentially the model's predictions based off of where it thinks that sample is from, purely from the microbiome. And it has not seen points, it has not seen that swab before, right? So it's not a case where it's like, oh, I've recognized this swab before, it's easily Pier 39 or something like that, right? But it was trained on swabs from around Pier 39 in previous time points, right? And so this is a situation, we've got kind of a variety of behaviors here, and we're still kind of teasing out what each of these different ones mean. So for example, we've got some real bullseyes, again, Pier 39. You can't see it, but the prediction points are right behind the marker right there. It's covered up there. My guess is sea lion microbiome, but I don't know. There's also a point down here where it strongly predicts down here by Hunter's Point, down here by Lake Merced, kind of Twin Peaks area. These are situations where, based off of the microbiome of samples collected from that region, the model is like very highly confident that it's from that particular area. We also have areas where it also shows some confidence, but it's like off by a little bit, and we're figuring out why, right? So here's a situation where samples collected closer down to the zoo, and it predicts Ocean Beach, definitely, but like a little further north, right? I wanted to chalk that one up to it being an edge point, but we're not quite sure. But again, it's still getting kind of the rough area of the city. There's also another one here at the entrance to Golden Gate Park. We collected one kind of right on this road right here, and it definitely thinks it's on the south end of Golden Gate Park, but it's a little kind of off and to the west, right? So we have these situations. We also have situations where there's a little bit more of a spread. We think there's potentially some sort of kind of like shared microbiome profile there. So it's like anywhere in here, right? So for example, there's those points. Oh man, we're doing so good. There's these points up by Richmond area, right? And there is a little bit of a horizontal spread there. We got these two different sets of points there. There's also another one with a very strong vertical spread, that kind of purple down there in the bottom. Our best guess there is that, the microbiome, at least in those top ones, it's like it can nail the latitude pretty well, but the longitude is iffy, and the other way around, right? And then we have other ones where it's like a little bit more of a spread, but it's like kind of in the right area, like especially these. It doesn't surprise me that it does a little bit worse in the downtown area because there's just so much going on here, right? But again, this is something that we're still exploring. The idea is there are pockets of San Francisco in particular, potentially due to microclimates, the fog, the type of people that live there, the type of activity that kind of characterize these different areas, right? So it's not like, you collect a dust swab outside somebody's house and you send it to a lab and then there's somebody knocking on their door the next day, right? We don't have that precision, and I'm doubtful that we would get to that particular stage because it depends on the particular biology behind this stuff. But there are certain spatial regions that you can kind of predict pretty well. And we did this in San Francisco. We also have it throughout the Bay Area and California, and you can pretty well pick out different cities and regions within that city based off of their microbiome profile. So this is the last application. The last thing I'm going to end on, because we're a practicum partner and we've got a whole lot of stuff to work on. I just wanted to describe kind of the data science skills in general. I know you guys are early days in your program, but the data science skills that I have found most valuable in this position. Again, this is an early stage startup. We raised Series A fundraising, closed in February, right? So we're still early days ourselves. But at least in my case, the most high quality skills so far have been data visualization. Definitely, all these different plots and stuff you've seen here is either me or one of the other data scientists on the team. So you got to be kind of comfortable making data visualizations for yourself to understand what's going on. You also have to make data visualizations for other people on your team. The executives or maybe the lab team wants to figure out why these certain samples are having trouble amplifying in the lab, and you need to visualize something for them. And you also need to make these visualizations for external parties, customers, potential investors, that kind of stuff, right? So I've done enough data visualization at this point that I feel pretty comfortable with it, and it's definitely something that I use on a regular basis. I typically use, not to grandstand, but R, ggplot, that's my favorite. All right, I use for most of the machine learning stuff, it's Python, but in like data exploration and especially data visualization, I can fly with ggplot, and it looks fantastic. I have nothing but good things to say about ggplot. The other thing, statistical modeling and machine learning models, right? We have to do some sort of data analysis, right? And so that's kind of where my training has been. That's my particular focus in our data science group. I'm one of 3 data scientists, and I typically do the statistical modeling part of things. But we all kind of work on the same projects, and so that's super important. The other thing that might go underlooked in a data science program, as opposed to maybe like a more hard stats program, is experimental design. I wish I had paid closer attention in my experimental design class, because what happens is we're always exploring new applications, right? And we want to enter these kind of like pilot projects and kind of understand if our technology might be valuable in a particular sense. And so that requires setting up some sort of experiment, right? To try and figure out, like, does this technology have does it have legs in this particular situation, right? And so you never usually get what you want. You can't design the perfect experiment, as we've figured out. But oftentimes you're working with other people, and you have to figure out, okay, I have this number of samples, and I want to answer question A and question B, and question C would be fantastic, but I'm probably not going to be able to answer that with the data available. So I'd prefer to get samples from this particular area and this particular area while fixing these particular other factors as best we can, and just kind of planning out these things. Of course, the second you actually plan something, reality hits and it quickly derails, and so you're kind of guiding this train on tracks that don't exist anymore. But it's helpful to have a plan at the beginning, right? So that's experimental design. The other thing is, if you're a data person at a small company, you're going to be leaned on for business intelligence and data-driven decision-making. Does it make financial sense for us to explore this particular market? I'm usually not involved in the market research, but I'll definitely be consulted with numbers and, essentially just this kind of, whenever there's some sort of data available, people want a discussion and they want potentially decisions that are driven by the data. And so that was not something I ever got any experience in academia, obviously, but it is an important piece, and that's something I'm glad I've gotten experience with. And the last one is, in this particular case, it helps to have some specialized knowledge of microbiome data, but my bigger thing is, it's really awesome to get experience with a variety of different data sources, but it always helps to kind of find a particular data niche and explore that a little bit more, right? So I still would not consider myself particularly knowledgeable in microbiome data. I've learned a lot at this company on how the data are generated, kind of different factors that might come into play about why it doesn't look the way that you want it to look. But it never hurts to, if you find a particular area and you're a data scientist that's interested in that area, there's always these nuances about the data that are very, very, very valuable to kind of drill down and understand, right? So I just encourage you to find whatever it is that you want to explore a little bit more. There's something special about that data that if you know that, you can kind of tailor your analysis appropriately. And that's not always easy to do, but it's super important. And that's it. So thank you very much for having me. This is my email at the bottom. It's pretty easy to remember. And like Steve said, we are a practicum partner, so hopefully some of you out there with some biological leanings might be interested. So keep us in mind. Yeah. Hi. Thank you.
I have a question. So one of the assumptions may be that the microbiomes are very stable over time.
Mm-hmm, mm-hmm.
When you did your research in San Francisco, did you find that, or are the microbiomes changing?
Yeah, no, great question. There are seasonal patterns, but they're stable within, they're more spatially stable than they are temporally stable, right? Which was key, right? But there are definitely seasonal patterns. So when we've collected samples in March and then March again, it's amazing how well they look like each other and how much they're slightly different from samples that we see in summer or fall. Yeah.
Can I do a follow-up?
You can do a follow-up.
We get the dust comes from the west.
Yeah.
Do you find any similarities with Asia dust or at all? Or?
If I had a rich database of Asian dust, I would certainly take a look. I mean, potentially, although, I'm not sure. But probably. Ooh, well, yeah.
So one of the problems we face in quality control departments for like drug manufacturing is we have to find like root cause origins of particular outbreaks.
Yeah.
And I just wanted to ask, what level of spatial precision do you get? Do you like within a building or within a set of buildings? It's like a very common problem.
Oh yeah. It depends on a lot of factors. This is the least satisfying answer, but I'm a statistician, so I get to say it depends to basically everything. We're like, oh yeah. But yeah, it depends. I mean, as you saw in San Francisco, we can get pretty good resolution to neighborhood. You're at the mercy of like how many samples you collect in a particular region. Another thing that might come into play to differentiate buildings is kind of what is their like HVAC system? Are things normally exposed to the outside air? Are they more internalized, right? And so it is possible that within a kind of a smaller scale, you could differentiate things. You might also get to a point where things look too similar to be able to explore that.
So if the data was a little bit more structured in that there are isolated rooms and stuff, how would you handle that?
I mean, if there's isolated rooms, that's a pretty good spot to be, because then you could get, set of samples from one, set of samples from another, set of samples from another, and then if it was a matter of determining which of those rooms it came from, right, you'd have a much better shot. But yeah, I mean, the spatial resolution is something that we're still trying to tease out. Thankfully, in the applications that we've looked at, it's not as though we need to go kind of knocking on this building versus this building, but it's certainly something that we're kind of excited to explore a little bit more. Yeah. Okay, I saw your hand. Yeah.
So when you have a trained model and you have like a sample of dust that you want to make a prediction on, how quickly can you make a prediction? Is it like days in the lab, or is it pretty quick?
That's a good question. It takes a while. I mean, once the data is digital, it's like that. But typically in the lab, depending on, the boring part is usually there's some shipping time associated with getting the sample there, depending on where it was collected, right? And then it's usually about like a day or two in the lab, and then like another day for sequencing, and another day for maybe processing. So it's about, if you were super on it, about a week delay. Obviously, we want to shorten that time period, especially for kind of these very time-sensitive ones. But for now, if we hustle, we can get it turned around in a week. But usually, we haven't had any sort of like ticking time bombs or anything like that. Yeah. Yeah.
So you showed those like stacked horizontal bar charts
Uh-huh.
with the phylogenetic breakdown
Yeah.
of the data that you got. How much of that do you care about in terms of like actually identifying what's present in the microbiome, and how much do you care about just finding the overlap that corresponds to that?
My answer would be, I usually just care about overlap. My coworker Simon, who's definitely has much more of a biological background, he got his Ph.D. in plant biology. He's very interested in who's there, not just that something's there that is associated with something else, right? And so there's certain situations when we're doing the kind of machine learning model prediction, it's not super important that we know kind of what these particular strains are, other than, hey, this one has these strains and this one also has these strains, so they're probably spatially related. But there's other situations like we saw with those stacked ones and these kind of spreads where people are interested in understanding just what's there, right? I don't need to do any sort of machine learning model prediction. I just want to know what's there and what that might tell us about kind of this particular environment. So it depends, in this case, on kind of what it is that you want to know. Yeah. Yeah. What's that?
Once you process the data and get it ready to analyze, what does the data look like? Is this sort of like a text file, and what are you doing on it? Is it kind of like scrambled bits of DNA, and you're seeing, oh, there's one, there's another one, or what is that?
Well, yeah, so at the beginning, it's just this massive text file of the DNA sequences, right? It also, in that text file, is not just the DNA sequences, it also is recording how, the sequencer records how certain it was that it saw, like, it says this is a G, but it says, like, I'm like 70% certain that's a G, right? And so we need to take that information and process it down to something that's usable, right? Preferably data points, if we're thinking in a tabular data form, data points, microbial DNA strands, and essentially within each row is essentially how many times I saw that particular DNA strand in that particular sample, right? So at the tail end of this, ignoring all the kind of extra metadata that's associated with the samples, we arrive at kind of a table. It's a pretty large table, and there's better ways to store it than a table, but you can think of a table where each row is a sample and each column is essentially a DNA sequence. It's associated with different microbe, and then the values in the table are the counts, the number of times that particular DNA sequence was read. So we have these essentially very high dimensional vectors of counts that sum to some total that's varies by the different samples, but then we can look at that with like what the composition is of that whole particular thing. So we get it down to a form that's much easier to work with than these like raw DNA sequences. Yeah.
So going back to actually understanding what's in the sample,
Sure.
it seems like you had some unwanted variants in there. So variants there where you want.
Sure.
maybe going back and actually understanding, like, it is sea life.
Yes. Yeah. Yeah.
It's, because these are real genomes,
Mm-hmm.
so you can class it against this databases and actually maybe refine or hypothesize.
Yeah.
So you really like doing the like molecular biology can help you tune up the data.
Yeah. Yeah. That's certainly something that we're exploring. Usually we're at the mercy of the databases that are available, right? There is pretty good databases of bacteria and fungi that people manage. As far as I'm aware, I've not found the celion.db file that I need. But I see your point. There's also, there is this natural variability, and we are working on better characterizing, like, what is irreducible, from a stats perspective, what is irreducible variability, and what is variability due to these other factors that maybe we just haven't considered. Yeah. Yeah.
You mentioned Bangladesh and Nepal.
Yeah.
There was then authorized workers.
Mm-hmm.
What do you do in an instance where you find something like that? What are the ethics around that?
Well, typically in this case, at best we would look at the dust that is on the product and whether or not it matches the facility that was approved to manufacture, facility or set of facilities, right? In that particular case, if there is not a match, right, that's something that we inform the brand of, and they have these partnerships with these companies, so it's not as though they're like, all right, snip, snip, you're out of here, But there's an opportunity for a kind of conversation there, right? Like, it's possible these places have facilities that the brand wasn't aware of, and they can kind of like, shake out the details there. It's a conversation that happens with our business team, and then the brand, and then kind of loop in these other companies. Yeah.
I had a similar question about the textile industry.
Yeah.
How is the fingerprint affected when I've seen products where it says like manufactured and made in the USA, but the materials are produced somewhere else.
Mm-hmm.
So when you find this fingerprint, like which one does it really capture, and does that like cause like a lot of issues when you're
To the point of understanding sources of variability, where the raw materials are from is definitely one of them. What we try to do is get, what does a non-manufactured raw material or microbiome look like versus the microbiome that's associated with the particular facility that it was made in. But you're right, that is certainly something that we have to factor into our analysis. Yeah.
Have you been able to distinguish between the two, or do you just categorize it as like a hybrid?
Right now, it's more kind of a hybrid sense. Yeah, we're getting better at that.
So the swabs that we are using to collect data, so each of them must have their own microbiome footprint, right? Because they are being prepared at different facilities and at different facilities.
Mm-hmm, mm-hmm.
So do you actually look for that, and how do you deal with that?
Yeah, they're supposed to be sterile when we get them, right? I got some chocolate, so maybe they're not. We have tested to see, like, are we starting from a kind of corrupt location or starting point, right? Like, if it already has stuff on it, that's bad, right? We haven't been able to like recover any sort of DNA off of these things. So if something is there, it is present in very, very low numbers. But yeah, I mean, I had to pick my battles on what I'm paranoid about as far as sources of variability that I don't want, and I'll trust the swab manufacturer that when they say sterile, it's pretty much sterile. Yeah. Yeah, boy, so many. I've went all the way through grad school, and I never got more than 5.
Let's say a product is shipped from city A to city B.
Yeah.
It makes 4 stops during the shipment.
Sure.
So using the microbiome data, you can tell me that these are the stops where the product was actually that it was checked or something happened. Right. So how do you know what route it took? Because you didn't talk about the time or how old the sample was.
No, that's a good point. I mean, typically when things are moving, it doesn't go through too many stops, and normally from the path, You're right. We don't necessarily have information that it was this stop versus this stop. But if the destination is over here, usually it's not this kind of crazy zigzag pattern. And normally, kind of our desire is to figure out kind of where it came from versus like where it ended up. And so in our situations, that the pilot with the packages being shipped around there with the multiple stops, that at least in our applications hasn't been kind of that's not usually the regular situation. Usually it's like it was here, it gets packaged, it gets sealed up, right? So think of it as like a little capsule, and then the next time it's opened is on U.S. soil, right? And so you're right. If we did want to kind of like, and this is something that we've talked about, which is like if you want to be able to follow something, we certainly need to devote time to figure out like how the temporal kind of stops affect the signature. Yeah.
All right, maybe we'll do one more.
Okay, so I already pointed at you. I'm sorry.
So what kind of models are you using once you have your table of reads versus samples?
I would love to tell you. I would say my co-founder would say it's proprietary. So, but it's sound statistics, I would say. I'm responsible for it. Yeah, we're using machine learning models. Sorry.
Let's thank Neal again.
Thanks, guys.